Hello, everyone, and welcome to today's webinar, The Impact of SARS-CoV-2 Genomic Surveillance Locally, Nationally, and Globally. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. There's been a slight change to our agenda today. I'd like to now welcome our first speaker, Dr. Jared Aulcair, Associate Dean of Professional Program and Graduate Affairs, Northeastern University. And joining him today, Dr. Nicola Normino, Director of the Cell Biology and Biotherapy Unit and Translational Research Development, National Cancer Institute in Naples. Jared and Nicola, welcome. You may now begin your presentations.
Great, thanks for that introduction. I won't belabor the title slide except to say that I've listed my email address here, and please don't hesitate to drop me an email if you have questions or want to talk about anything further. Northeastern is a wonderful university that's all about collaboration and partnership, so definitely feel free to do that at any time. Of course, just briefly, I'm going to show the disclaimer slide. I won't read it to you because we've all seen these slides many times, but this is just the disclaimer. All right, so first I thought I'd start by telling you a little bit about Northeastern University in Boston, Massachusetts. Oftentimes when I travel and say Northeastern, people are like, you mean Northwestern? No, I mean Northeastern University in Boston, Massachusetts, in the great state of Massachusetts. We're a top 50 university. We are an R1 research university, according to the Carnegie standards. Our career services has been ranked #1 several years running. We are a global campus that has a global network of campuses in Boston, in Burlington, in Portland, Maine, San Francisco, internationally in London and Toronto and Vancouver. Our claim to fame, if you're familiar with Northeastern at all, is experiential education. So our signature co-op program, youth-inspired, work-inspired research and training is at the heart of what we do. And we have approximately 5,000 faculty and staff and 28,000 students or so. In the context of what Northeastern doesn't have, which is particularly important to think about in the context of what I'll talk to you about today, is we have no medical school or clinical labs. In our history, we had been considered a commuter school until maybe about 20 years ago when we really pivoted to a top-notch research and education institution, but still no medical school or clinical labs. We do have the Bouvet College of Health Sciences that does nursing and other clinical activities, but no clinical labs or medical schools. So what I thought I would do is start by telling you the journey of how a university with no medical school pivoted to respond to the COVID-19 pandemic in 2020 and how it's evolved since the program started so many months ago at this point. Feels like an eternity, but really it's only been, what, about a year. So briefly, I broke it down into a sort of a timeline of events, and I'll break these down as I go through the presentation slowly, but you can see that, just like the rest of the world, on March 13th, the university decided to deliver all of its educational content fully remote. This day will always stick out in my mind because that week of March 9th, I was providing a training program to a number of international regulators from Africa and kept thinking to myself every day, please don't close the university because I have to finish this program and get these folks home. We did finish the program, and luckily, they were able to get the last flights out of the airport and back to Africa. And the second the university closed, a couple of weeks later, we sent home all of our students and then started planning for the reopening in fall, right? We wanted to make sure that we opened safely. And that entailed a number of different steps along the way, instituting a reopening task force. And then what I'll focus on here is exactly the result of what that task force decided, which was to start our own testing lab. Back in the day when they asked me if I would help them do this, I thought it was like everything else in my life. I'm naive and said, yeah, no problem, we could do that. That's going to be easy, which of course it was not. So on March 13th, like I mentioned, we went to remote instruction. A couple of weeks later, all university students and employees were fully remote and not on campus. Research continued throughout the whole entire pandemic, which I think is important to note, especially that related to SARS-CoV-2. And then, so in essentially 2 days, everything that was happening at the university was converted to online, which is, as you know, both rapid and necessary. So then, the next few weeks, the university formulated a reopening committee, which was made up of the senior leadership and other key stakeholders. That reopening committee was monitoring the current situation in Massachusetts and across the globe, since we have campuses everywhere. And out of that reopening committee came a testing task force, which is where my initial role came into play. So from the very beginning, the testing task force had a four-pronged strategy to look at best options: partner with large diagnostic labs to do testing, monitor new technologies, partner with other academic institutions, or set up our own testing lab. We realized to open safely in the fall of 2020, we would have to have testing as part of our daily activities, or what the norm would be for this academic year. And these are the four things that we considered in the process. We learned pretty quickly that partnering with large diagnostic labs was going to be a challenge. The turnaround time at the time, this point in time in April of 2020, was variable. The Medicaid was dictating the price of what those tests cost, and would only allow for one test per person to be paid for, so that limited our options. So that was some of those challenges there. Thinking about new technologies, one of them in particular that we monitored for testing was next generation sequencing, which I'll talk about in a little more detail, but how we use that now in a little bit. But at the time, there was no FDA approved EUAs for NGS, and the timeline and scalability was unclear. So those are a few challenges around NGS as a new technology. We did monitor a number of other new technologies, such as antibodies and antigen tests. Again, they were a little bit further behind. We did look at partnering with other academic institutions in the Boston area for testing. Most of them had medical schools. So Boston University has a medical school. The Broad Institute has clinical labs and is part of Harvard and MIT, et cetera, right? And as Northeastern had no medical school, we were in a unique situation. So we didn't partner with any of the big schools because they were all doing their own thing. We did consider helping some of the smaller schools around us, but in the end, weren't able to do that. So then we decided that the best approach with the least amount of challenges and risk was creating our own testing lab, which is the Life Sciences Testing Center. And from Memorial Day until July 12th, we had further discussions about whether that actually should happen or not. Was it a good return on our investment? And on July 12th, we got the final green light to start processing paperwork and get the lab set up. While we were doing that, obviously, like I mentioned, one of the things we had to consider was what type of test to institute. We were only considering at the time molecular diagnostic or PCR-based tests or serological testing. We, of course, decided to use molecular diagnostic testing because it is the gold standard in testing and most sensitive and most specific. So that's what we decided to do. Excuse me. The Life Science Testing Center was born on June 12th, like I mentioned. We have no medical school or clinical lab, so I was starting from scratch. It was me who was asked to get the center up and running. We had to decide where to locate it. And if you've ever been to Boston, you'll know that space is limited on the Boston campus, in Boston in general. We have a regional campus in Burlington, Massachusetts, which is about 20 miles northwest of Boston. I had a lab there already that I had just finished construction on new lab space. Okay, I didn't do the construction clearly, but the construction on the new lab space had just finished. So I had 4 walls that were set up to be a lab that had nothing in them. We sort of hijacked that space to get the testing lab up and running, which was about 3,900 square feet or so. We hired South Shore Lab Consultants, a great consulting agency that helped us set up our CLIA lab by providing standing operating procedures and a number of other things, including vetting CVs of employees to make sure they fit all the guidelines that we needed them to. For me, when I think about testing, when I think about anything that I do, at the heart of it is the patient, which in this case would be our student body or employees, and ensuring quality and accurate results. So quality and accuracy are the foundation of everything we do. So with that at the foundation, we determined what kind of test to use: nasopharyngeal, anterior nair, saliva, or other. We had to acquire the appropriate equipment, hire and train the staff, select the laboratory information system, which one would have thought would have been easy, but was much more complicated than I hoped. Determine the sample flow, secure the testing supplies, the consumables and disposables, do the validation, and get certified. Obviously, supply chain was crunched for most of these things at the time. That supply chain crunch doesn't exist so much anymore, except for maybe in the plastics, but that was a huge challenge at the beginning, and this was all a challenge that we faced. One of the things that was super important to me was picking the right test, right? I already told you we were going to go PCR, but what kinds of tests were we going to use? I looked at the CDC test. I wasn't, and I'm still not a huge fan of it. It monitors one gene on the viral genome. It largely had been only the N1 primer. The N2 and N3 had been set aside, although N2 is being used more routinely again. So, to me, that's not a good N. There's not enough reproducibility, if you will. So we looked for tests that would allow us to monitor several different genes. I did my PhD research in a drug resistance lab, so for HIV, so I understood very well how RNA viruses mutate and evolve, clearly knowing that they want to remain in existence and not kill their host. So from the very beginning, I was concerned about variants and what that might look like. So I wanted to monitor more than one gene across the genome, which is why we landed on the TaqPath COVID-19 combo kit, monitoring ORF1ab, the S gene, and the N gene. And the S gene will be an important thing as we go through some of the later points that I'll make. Sample workflow, just like anything else, we set this up in June. Sample collection, sample prep using liquid handling robots, PCR, and data analysis. Nothing special there. So from June 12th or so to July 31st, about 6 weeks, we set up the lab, installed all the instruments, did all the qualifications, hired the staff, went through the whole process, and on July 31st, had our inspection by the state of Massachusetts and our original CLIA registration, and were approved for processing tests that day. The testing sites were set up. Those were sample collection sites on our Boston campus, one for asymptomatic patients. So the Cabot Testing Center was for routine surveillance testing or medical screening. We test students every 3 days and employees twice a week if they're on campus, 5 days a week. The Huntington Testing Center was a symptomatic site. I can tell you I learned a really great example. All of our modeling had suggested that we would be receiving 200 samples a day on our first day of actually processing patient samples, which was the middle of August. And at 1 p.m., 1,300 samples arrived on our doorstep, which we were not ready for. I wanted to scale hundreds to thousands, not thousands in the middle of the day, and we were closing at 7 p.m. So, that was definitely a challenge we had to overcome, and luckily we did. So this is what we landed on, AN swabs. The samples are transported from Boston to the lab. All of our tests are medical tests ordered by a physician. When students return to campus, like I mentioned, they were tested on day 1, day 3, and day 5 before they could go back to class. And then they were tested every 3 days. That continues through the end of this academic year, so May of 21, the first week of May of 21. Employees are tested every 5 days, and the testing is for students, employees, and contractors who are embedded in the university. So at the time, we had these equipment, just highlighting quickly, 8 biological safety cabinets, 7 liquid handlers, 9 qPCR machines, approximately 75 clinical staff. I did realize we needed support staff because, like I said, we have no medical school or infrastructure. So we brought on a lab manager a supply chain manager to handle the supply chain. I was spending a lot of my time dealing with that. QA/QC to ensure the quality of everything we did. IT, lab information manager, and a business manager. And we went from operating 12 hours a day to 24/7, 365. Shortly after that, we expanded the lab space another 1,300 square feet and increased our capacity to about 15,000 to 20,000 samples per day. We also did receive IRB approval for research, so we have a research repository and registry. We want to understand COVID-19 and learn how we might prevent the next pandemic. It's also a really interesting thing as I'm sort of talking about the Northeastern testing program, which is sort of a local program and really can give us a proxy for what might be happening in the community around Northeastern in Boston, but also really highlights the need for in my opinion, a larger scale network of infrastructure for testing moving forward. So as I mentioned in the expansion, we doubled our size and doubled our instrumentation, and then started to consider testing beyond Northeastern to use some of that capacity if it was possible, which it was. We started doing K-12 testing just about mid-March, beginning of March, to help the primary school kids get back to school. So looking at our 4-pronged strategy, partnering with large diagnostic labs didn't work out. We still continue to monitor new technologies, and we'll consider partnering with other institutions and clearly have our own testing lab that's operating, and are thinking about COVID testing and other tests we might do in the future and research opportunities. We are transitioning to pool testing over the next month or so. We're looking at how we might support serological testing and other sort of vaccine passport related tests, including, we didn't really have a flu season this past year because of mask wearing, which is great, but one would anticipate having respiratory diseases increase in prevalence in the population as we go into the next flu season and common cold season. So we might have a bunch of symptoms and we don't know what disease it is. So we're looking at combination tests like flu, COVID, and other respiratory combo kits. So briefly, I just want to transition to the phase 2 of our timeline, which was in January, right? And that's where we started, the vaccines started coming on board or online in December. We started hearing much more about variants and started to determine how we were going to leverage our testing program to monitor those variants of concern, both from an isolation and safety standpoint, as well as potential information that we could learn by the large number of samples that we collect. So the population began to get vaccinated, like I mentioned, and we started hearing about variants of concern. I just listed a few of them here that we had thought about from that January timeframe and continue to think about, like the UK variant, B117, B1351, the South African variant, P1 in Brazil, and others that we monitor currently to see what's happening, right? One of my things as we move into the summer and the fall is more people become vaccinated as the university set up our testing infrastructure, that we continue to monitor vaccinated people if at all possible, so that if variants of concern start to become predominant for whatever reason, especially as international students come back in fall of 21 for the next academic year, we're prepared and can be proactive instead of having to be reactive. So I think it's an important thing for us to monitor. Also could add tremendous amounts of data and value to understanding the evolution of disease variants in populations and how we might prevent it. So we do have next generation sequencing as part of the infrastructure here at Northeastern. It's in my Biopharmaceutical Analysis and Training Lab, so not technically part of the testing center. Variance, caused us to develop the sequence strategy, obviously. Like I mentioned, we use the SARS-CoV-2 TAC-PATH kit, which has ORF1A, 1B, N gene, and S gene. We started seeing a large number of S gene dropouts, which we had predicted and hypothesized were due to B117. So we decided to sequence those positives in collaboration with the State of Massachusetts Department of Public Health and on our own. And over a 3-week period in March, we were seeing about 5% of our samples being S gene dropouts, which was confirmed by sequencing. And over the next several weeks, that 5% went to 50%, that went to 85%, confirmed by sequencing and other variant panel use. So we saw the number of our positive cases increase drastically as B117 circulated our population. And over the last several weeks, or since then, those numbers have come way down, and our positivity rate right now is less than 0.1%. We get, far fewer positive tests, which also is a reason to remind people it's important to remain vigilant as time goes on, because the summer, of course, with windows open and people outside, we would expect fewer infections anyways. So currently we're using a multi-pronged approach to our testing protocols. The traditional testing, where we can identify a positive or negative using the qPCR that we've been using all along, sending S gene dropouts to a PCR using variant panels to identify specific variants quickly, and then sequencing those using next generation sequencing, as well as sequencing all of our positives, new and banked, to see what exactly the sequence of those viruses are. So this is currently what we're doing in a multi-pronged approach to keep our community safe and to understand more about the SARS-CoV-2 virus and pandemic. So briefly, I think I learned a lot of things over the last year. It's been a really exciting and stressful time setting up a CLIA-certified lab in 6 weeks, as you know, or might imagine. But I would really promote continued testing, especially of unvaccinated people, but also of vaccinated individuals, to identify vaccine escape variants so we can be more proactive versus reactive, like I said. We have seen 3 vaccinated individuals who are fully vaccinated, so 8 weeks post their last vaccine shot, that tested positive for COVID. One of them was tested outside of the university, so I don't have access to that sample. One of those samples was recent, and we're currently doing further analysis on that using the workflow that I showed previously with the variant panels and next generation sequencing. And one of those we analyzed in an initial round, and we're able to determine that the variant is either P1 or B1351. We're doing more analysis to suss that out. Next generation sequencing to me is crucial in identifying new variants. The ones that I know about concern me less than the ones that I don't know about in terms of variants, so I'm really interested in learning what those are. There's an opportunity for research, how we can really help progress next generation sequencing into a routine diagnostic tool, for example. So it's faster, it's reliable for sure, but faster and more robust. In the early days, one of the reasons we didn't consider NGS is the turnaround time was a little slower than we could do ourselves, but I think crucial. And sequencing in a pandemic, but sequencing of viruses in general, I think is an important thing to do. When we talk about the United Kingdom being about 10% are sequenced, and the US is about 1%, and that's awful. 10% is definitely better than 1%, but I think in general we can do better. I'm really hoping that we can drive that forward and look forward to automated systems helping with that I know exist. And the last thing that I really want to say is, lots of institutions invested lots of time and resources in setting up clinical labs and setting up different testing and diagnostic resources. From a national perspective, it's really a time to think about what a national testing infrastructure or pandemic response might be, or you could even think about it if you want to loop it into the category of sort of biosecurity in general. That doesn't mean warfare, right? But I'm talking security against pandemics and other things that might happen. So it's our opportunity for some sort of national infrastructure to be set up and maintained, clearly not at the level that it is, but maintained through partnerships with individual universities and other entities, spearheaded through large companies and government, so that we have nodes in a network that can help respond in the future. And with that, thank you for your time and attention. And like I mentioned, please feel free to email me if you have any questions or comments, and happy to answer a few questions now.
Well, many thanks for inviting me to give this talk on molecular pathology activities in the COVID-19 era. These are my disclosures, and this is an additional disclaimer from Thermo Fisher Scientific. Well, in the first part of my talk, I will briefly describe you the impact of COVID-19 pandemic emergency on cancer research activities in Italy. Then I will discuss some results from the literature related to the activities of molecular pathology labs during the lockdown. I will try also to show you some possible solution in situation of reduced personnel in the lab. And finally, I'll touch a little bit on the possible role of molecular pathology labs in the sequencing of the SARS-CoV-2 viral genome. And let's start to see first what happened in Italy in labs involved in cancer research during the lockdown. As you might know, in March and April 2020, in Italy, we had almost complete lockdown of the country. And in May 2020, the Italian Cancer Society launched this survey in order to assess really what happened during the lockdown in the labs involved in cancer research. The survey was completed by 570 researchers belonging to almost all European regions. And the first question that we asked to the participant is what happened in your lab? Did your lab suspend the research activity during the COVID-19 emergency? And actually 48% of participants declared that the activities were completed suspended, whereas the partial suspension was reported by 36% of the participants and no suspension from 16% of participants. Of course, the main impact of lockdown was on face-to-face research activities that was significantly reduced in most of the cases, and this was in part compensated by the use of smart working, and still now we are using smart working in some Italian labs in order to reduce the number of researchers that are present in the lab at the same time, thus reducing the possibility of spread of the virus. In the lockdown, about 30% of Italian researchers were involved in a research project that are related to COVID-19, and approximately the same fraction was also involved in other activities like testing swabs or other that were also related to the COVID-19. Thus, underlying, the willingness of Italian researchers to help in a situation of emergency like the pandemic that we are still facing. Quite surprisingly, after the lockdown, almost 20% of the researchers did not resume yet their activity, and even more surprisingly, in 28% of the researchers that returned to work were not offered any serological test or swab, whereas 21% of the researchers were not informed about. This means that in almost 50% of the cases, no contingency plan was prepared in order to ensure that researchers could go back to work in a safe environment. And because we are still in a period, in a situation in which the virus is spreading, we still have the high level of presence of infection in our countries. Well, I think that these data are real data on which we should think about. Let's see now what happened to the activities of molecular pathology labs during the lockdown. In this respect, we have some conflicting results coming from the literature. This, for example, is a survey that was performed in several European thoracic pathology labs, and what was reported that when the activities of March-May 2020 were compared to the activities of March-May 2019, only a slight reduction in NGS in the clinical diagnostics was observed. Slightly more reduction was observed in liquid biopsy and in NGS testing for clinical trials, but a clearly more significant reduction was observed for some specific tests like PCR or fish analysis. Now, these data are somehow in contrast with this report that describes a similar survey in a number of Italian labs involved in molecular pathology. What was reported in this study is that the number of tests really did not reduce significantly. However, there was a clear shift from NGS to more simple tests such as IDILA in molecular pathology labs, with also a reduction in the turnaround time from 7.9 to 5.3 days. So what happened in these labs? I mean, what the authors discuss is that in many cases, because of the reduced number of researchers, reduced number of personnel present in the labs, then the labs prefer to switch from more complex tests to more simple tests. Well, I have to say that in our lab, we had no reduction at all our activity of clinical diagnostic, clinical research, and these are the data for clinical diagnostics. The number of tests that we performed were almost the same or slightly increased in 2020 as compared to 2019. And actually, we continue to use NGS, but this is also due to the fact that we work in academic centers. We have a lot of clinical trials available, and of course, we have a request from our colleagues to test patients for a number of genomic alterations that offer the potential to enroll patients in clinical trials. This is just the example for lung adenocarcinoma. Actually, as you may know, the European Society of Medical Oncology recently released a recommendation for NGS testing in solid tumors, and ESMO recommends that in the clinical practice, you should use NGS for lung adenocarcinoma, cholangiocarcinoma, prostate cancer, ovarian cancer, carcinoma, non-primary, and a few other applications. Actually, what really ESMO recommends is to use NGS panels that are limited to genomic alteration that are level 1, so basically use genomic alteration that, for which there are approved treatments. However, in the centers that have clinical trials available, it is recommended to use larger panels, because if you use larger panels, you can identify patients that might be enrolled in a clinical trial based on genomic alterations. Still, if there is a reduction in the number of personnel in the lab, it might be difficult to continue to test for NGS all patients, but a solution in this respect might be represented by automation. And I have to say that the Genexus system might represent really a very good solution for clinical research. In fact, this is a highly automated system that goes from nucleic acid purification to library preparation, variant sequencing, variant interpretation, and reporting. Now, the Genexus is a very fast system, very easy to use, with a very limited hands-on time, and I think that this represents a very good solution to ensure an increase in the level of automation in our labs. Now, there are many panels can be used on Genexus. The Oncombine Precision Assay is a panel that is, according to my thinking, very good for clinical research. In fact, this panel covers 50 genes and all different types of genetic alteration, these 50 genes, DNA hotspot mutations, covenant aberration, as well as fusions. Now, this panel, of course, is based on the AmpliSeq technology, and if you use an AmpliSeq technology, you are limited to identify only known fusion, because you must know the sequence of the kinase and of course of the partner. However, the OPA assay has this exon tiling imbalance assay that allows also the identification of unknown fusions, and at least this assay is available for a number of targets that are listed here. In the next few slides, I will show you the results of this multi-center study for the evaluation of the Oncomine precision assay on GeneXus. This study was run in 4 different labs: Porto, Basel, Charité in Berlin, and my lab in Naples. Each of these labs selected a number of samples that were already characterized by using different techniques. These were either FP or plasma samples, and these samples were tested for a number of genomic alterations. This slide shows the types of samples that we tested, most from lung cancer, but also colon cancer and other tumor types were tested. The cellularity of these samples ranged from 10% to almost 100%. These are the variants that were identified in this study, in the samples that we tested, and all different types of study, in all different genes, and here are shown some actual genomic alterations that are relevant for therapeutic intervention. As shown in this slide, we could identify variants at different allelic frequency, and also variants at allelic frequency as low as 2.79%, thus confirming the high sensitivity of the AMP-seq technology. When we compare the results of the OPA assay with the previous testing, the concordance range between 94 and 100% with a specificity of 100%. So I would say very good results. Quite a few copy number variation present in samples that we analyzed, and they were detected by the OPA assay, but also confirmed by previous testing. And more importantly, we could identify in this sample 29 different isoforms, fusion isoforms. And as shown here, we identify fusions of FGFR, ARC, BRAF, RET, NTRK, METAX, or for skimming genes, all different types of fusions. Among the different fusions, we could also identify a novel ARC fusion. This fusion was identified thanks to the imbalance assay, and it was confirmed by using FISH assay. The concordance with the previous test was 96%. And finally, we had also the chance to test a number of plasma samples. These plasma samples contained mutations at almost 50% of these plasma samples contain mutations at a allelic frequency that was below 5%. And as shown here, all different types of mutation were identified. And when we compare the result of the OPA assay with the previous testing, the concordance was between 95% and 100%. So in conclusion, I mean, these results suggest that the GeneXus and the OPA show a very high performance in terms of variance identification. The concordance with the previous test was very, very high. More importantly, I would say that the implementation of the OPA on GeneXus was successful, was very easy in all 4 labs, and we are very happy now to use this workflow in our clinical research. And last few slides, I want to discuss on the possibility to sequence the SARS-CoV-2 viral genome for monitoring the evolution of variants. Now, I'm not a virologist, but I think that everyone knows that one of the issues that we need to face is the evolution, the genomic evolution of the SARS-CoV-2 virus, evolution that is always ongoing because of the huge spread of the infection and also because of the slowness of the vaccination campaign. Now, some of these new variants might be of clinical interest because they might be associated with the increased ability of the virus to spread, or to increase resistance of the virus to antibodies or vaccines. Now, Thermo Fisher Scientific has available this panel for SARS-CoV-2 genome sequencing. This panel contains, actually this panel is formed by 2 pools with 247 amplicons, allowing over 99% coverage of the SARS-CoV-2 genome. And this panel, of course, can be run on different sequences, the S5, but also the GeneXus. But if you use the GeneXus, the advantage is that the workflow is very simple and very fast. So by using GeneXus, it might be possible, to test the samples in a few hours or in less than 1 day, and this might be very important to monitor the evolution of the virus in particular situations. Now, we wanted really to test how the GeneXus was able really to sequence the SARS-CoV-2 genome, and for this reason we ran this small study in samples from 27 patients. These patients were from the Campania regions, and the swabs were collected in March, April 2020, so in the first phase of the pandemic in Italy. And as shown in this slide, these patients had variable patient clinical conditions from asymptomatic to intensive therapy unit. First thing that we did was to quantify the viral content of these samples by using reverse transcriptase PCR, and we classified these samples in two groups: 15 with a high viral titer, more than 200 copies per microliter, and 12 with a low viral titer with less than 20 copies per microliter. The samples with the high viral titer were sequencing, used the SARS-CoV-2 research assay, whereas the samples with low viral titer were sequenced with the SARS-CoV-2 low titer research assay on the GeneXus. These are the results. The complete genome of the SARS-CoV-2 was successfully obtained for 21 out of 27 samples. I have to say, however, that in additional 3 samples, we had an over 95 coverage of the SARS-CoV-2 genome. The complete genome was fully sequenced for all 15 samples with high viral titer. For the 2 samples with the viral genome copy number less than 200 but higher than 20 copies, and also for 4 out of 10 samples that had a very low viral load, less than 20 copies per microliter. In the 21 samples with the complete genome reconstructed, we found 51 variants with the 29 non-synonymous mutation, and of these 29 non-synonymous mutation, 7 were in the spike. According to phylogenetic analysis, 17 of these genomes belong to lineage B1, whereas 4 sequencing belong to lineage B1.1, and I think that these data are in agreement with the types of virus that were diffused in Italy at the time in which the swabs were obtained. We are continuing, however, to test patients in our institute, and by doing this a few weeks ago we could identify for the first time in Italy this variant B1.525 that has been already described in about 100 cases in other countries around the world. This variant is of particular relevance because it contains a number of mutations, including the spike E484K mutation, which has been associated with resistance to antibiotics and vaccine. And of course, now we are monitoring the diffusion of this variant in our region. So my conclusions. I showed you how the COVID-19 pandemic significantly affected the activities of research labs as well as molecular pathology labs. I do believe that automation in molecular pathology labs can be of help in conditions of reduced personnel, and in this respect, the highly automated GeneXus system can really simplify laboratory workflow and reduce the turnaround time. The data that we collected in our study together with colleagues in other centers suggests that the open panel on GeneXus really can guarantee an adequate genomic profiling of cancer samples. And finally, the rapid sequencing of the SARS-CoV-2 genome with the GeneXus may facilitate the monitoring of the evolution of the virus. My very last slide is to thank my collaborators, the colleagues at the other BTS site, Basel, Berlin, and Porto, colleagues at Istituto Superiore di Sanità, Luca De Sabdo, Gabriele Vaccari, who collaborated in the project on the SARS-CoV-2 genome testing, and finally, colleagues of Thermo Fisher Scientific. And of course, I thank you all for your attention.
Thank you, Chair and Nicola, for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's take a look. Lots of great questions coming in. Here's our first question, Jared. What specific learnings would you share with other academic institutions interested in establishing a campus-wide testing and surveillance program?
Excellent. Great question. I would say first and foremost, do your homework and pay attention to what's happening around you. The first step for us was setting up the lab to identify positive and negative patient samples, and then to integrate that with next generation sequencing as we move forward. In the pandemic, genomic surveillance is going to be more important and more predominant. So, setting those things up at once, and then really it's a great exercise to keep a campus safe and making sure that the campus can remain open. Our testing currently will also include vaccinated people, which I think is an important thing to monitor, at least as we go through the fall semester. So, we're doing that also. I mean, I set up the CLIA-certified lab in 6 weeks. I wouldn't recommend that. That was uber fast and stressful. But I think taking advantage of what others have done before you at this point is the best way to go.
Great, thank you. All right, is Northeastern treating individuals with specific variants any differently than others that test positive?
And again, another good question. Right now, we're currently not treating individuals who test positive for a given variant any differently than our other patients who are tested positive. Interestingly, of course, as we all are, we're monitoring closely the recommendations from CDC and elsewhere, and do see there is some data that suggests some of the variants have an infectious period longer than quote unquote what people might consider to be wild type. So those are part of our conversations in terms of isolating those people for a longer time, perhaps. But as of right now, we are not treating them any differently.
All right, thank you. All right, your next question, which area did you find difficult to handle as compared to others?
Yeah, that's a really good question. So I think one of the challenges for us initially, right, is a university without a medical school and now without any experience was one getting a research-oriented university to understand what it means to function within a regulated environment. We can't just change things overnight, obviously. That was a challenge. Another challenge we had was sort of volume, right? We originally, when we're starting to plan, we were thinking we'd process 2,000 tests per day, and that number drastically increased to max on our busiest days 7,000 tests per day. Now we have capacity for 20, so we solved for that, but that was a challenge. And then, we should have thought about integrating genomic surveillance from the very beginning of the process. I mean, I was thinking about mutations from the very beginning, but was inundated with setting up the CLIA certification itself. We should have integrated that much earlier so that we would have been further along in sequencing. We instituted that sort of in the January timeframe or mid-January timeframe. So we should have thought about that before and done that. But hindsight is 2020, and we're doing it now. So I guess that's all that matters.
Awesome, thank you. All right, looks like we have time for one last question. How are sharing sequencing data with public health organizations?
Yeah, that's a great question. So we share our sequencing information, broadly speaking, we share our genomic surveillance data, our sequencing data with the Massachusetts Department of Public Health. We have a great line of communication with them. Because most of our samples are from Massachusetts, obviously, since that's where the campus is, or all of our samples are actually from Massachusetts. So we have an ongoing dialogue with them and share that information with them. We also, when we can, send them some samples of sequence on their own genomic surveillance platforms just for confirmation so that we can check each other. So really great interactions there with the Department of Public Health in the state of Massachusetts for us. I'm sure the same elsewhere, that allows for these things to be monitored at a state level as well and a national level.
All right, well, thank you again, Jared and Nicola, for your time today. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.